Chubb and Christian Herrera, who have been integral in the project. They're not authors on this paper, this talk, because they actually haven't seen any of these results and don't know about it, but we expect that when we find the right people, they will be in them. Today the talk is about visual perception and the role that is played by opsin gene polymorphisms in the expression of retinal mosaics and how that affects color perception. And we all know that normal human color vision has this phenotype, 3 types of cone classes in the retina: small, short wavelength sensitive, medium wavelength sensitive, and long wavelength sensitive. We also know that the long and the medium wavelength sensitive cone types are inherited by way of X-linked recessive types of mechanisms, so that a mother who carries a polymorphism or a mutation passes it, is obligated to pass it on to her son and may pass it on to her daughter. We also know that there are some of these polymorphisms that give rise to pretty significant shifts in spectral frequency of the particular photopigment classes that are expressed. 5 nanometers might not seem like a big difference to you, but if you consider that L and M peaks are only separated by 30 nanometers, then 5 nanometers may seem a little bit more significant. We know a lot about the types of phenotypes that arise for the top 3 panels, but we know much less about how the color perception that arises in the bottom panels. A particular polymorphism that is very interesting for visual perception is this one polymorphism that exists at codon 180 of the M and L opsin genes. Substitution is a single nucleotide polymorphism substitution. 1 protein shifts and you get a 5 nanometer shift in spectral frequency of the cone type, and that's pretty prevalent in the population. A question that has been asked is what kinds of tests might reveal a potential tetrachromatic difference if one exists. And it turns out that standardized color vision tests that are out there have been woefully disappointing in terms of being able to demonstrate whether or not a potential tetrachromatic difference exists when you have a person with a genotype with a genetic potential to express all these cone classes. So what we've done and what I've done in the past is publish results using kind of non-standard, kind of innovative ways of looking at this, not using things like Maxwellian view types of human circumstances, but looking at more naturalistic human circumstances, because the idea is that that would optimize the chance to detect a difference if one was there to get. And so in this particular project, we asked whether or not we could use something like a minimal motion phenomenon in order to assess whether or not there are differences in the types of perception that you get across these different genotypes and phenotypes. Minimal motion phenomena is an established paradigm that's very reliable and sensitive. Anstis and Kavanaugh originally published on it, and then Sperling has done quite a bit, as well as Chubb. The actual stimulus looks something like this. You have a successive set of frames that are presented. This is in time here that we're showing these panels of 8 frames of gray and colored types of stimuli. The frames are presented flickered at about a 14 hertz rate, and Winkler did some work on this across different color spaces. And the idea is if there is a brightness match between the gray and the color portion of these frames, then it produces an illusory motion that spins in kind of clockwise direction. And if there's a brightness match to this darker gray that you see in these frames, then it turns out that there's an illusory motion that spins in a counterclockwise direction. Okay. The results I'm going to show you today are from 4 female subjects. 2 of these female subjects are carriers of opsin gene polymorphisms. The first one is a carrier of an L-cone heterozygosity and deuteranopia. JK is a carrier of polymorphisms on both M and L opsin genes. And the bottom 2 subjects are normal trichromatic subjects. We presume that the retinal phenotypes of these subjects cluster into two groups, tetrachromat and trichromat, because there's much evidence that if you possess the genes for these opsin polymorphisms, then you actually end up expressing more cone classes in your retina. And it turns out we also have another additional variable, which is that some of our subjects are professional artists. They make money painting and selling their paintings. Okay. All these subjects tested normal on our standardized color vision assessment test that has no deficiencies. Okay, questions that we can ask are, for example, this kind. Independent of our training, is photopigment genotype an important consideration for MM-isoluminance settings? So would we find that potential tetrachromats are different from potential trichromats? Second question we can ask is, does the life history of our training seem like an important factor in addition to photopigment genotypes for MM-isoluminance settings? So we can ask whether or not a trained artist who is a tetrachromat looks different from a trichromat who is not a trained artist. The backstory to this second question is that we believe perceptual learning is an important prerequisite for the realization of functional tetrachromacy. This is not an angle that has been discussed at all in the literature, but I think we found from our experience looking at these subjects is that it's not sufficient to just have a genotype or even to have the phenotype. What you need to do is you need to, from the starting point of being a child, have a deep training of working with color. And there's a lot of similar types of things like this in the psychological literature. And what may be the case is that the person who is trained as an artist, who has the genotype, ends up being what's called a strong tetrachromat, that is, she's fully functionally tetrachromatic. The other individual who is a tetrachromat but not trained as an artist is a weak tetrachromat, which means she expresses the four cone classes in her retina, but she doesn't actually have the strong level of color discrimination that may differentiate her from a trichromat subject. Of course, there's a whole gradation of possibilities in between there. So, okay. And of course, we can also find that there is no effect on MM-isoluminance between these different types of considerations. Okay. Consider the plane of equiluminance that's shown in this particular CIE lab model. The middle plane shows you the hue circle and the area of color space where all the colors have been perceptually equated to be the same perceived luminance. Okay. The stimuli that we use sample from one of these planes in space. We picked 20 colors around the hue circle, which were chosen to be highly saturated within some constraints of the RGB monitor that we were using. And in the 20 conditions, in the MM-isoluminance task, the way that the variation occurs is that the subjects, the stimuli can vary along lightness directions that are orthogonal to this isoluminant plane. And it turns out that settings are established as average positions where the motion reversal occurred in the MM-isoluminance task, and I'll show you a kind of mock-up of that in a minute. So these two red X's may be places where, for these two colored loci on the hue circle, the subject chose a lower luminance setting for this greenish stimuli for MM-isoluminance, but for MM-isoluminance for the blue one, needed a little bit more luminance. Okay? And it may be the case that two subjects may arrive at different levels and show different patterns of settings. Okay. To the degree that these settings arise from first order motion based on luminance, they tell us something about the luminance contributions of cone classes that respond in the different regions, the spectral regions tested. And this is something that's really elaborated in a technical report that is being posted pretty much as we speak. So one further wrinkle that we did on this design was to optimize the chances of detecting a difference. We chose a lower gray adaptation level than is typically used in this particular type of experiment, because we found that in this one candidate potential technocrat's artwork, she tended to represent a lot of color in the low light conditions and shadows of her paintings. So we thought, well, we'll test a low light condition as well. And we also, so that's the mock-up of the design. And we also chose an orange adaptation background because of something we noted in a singularity in her sorting of the FM100 test. Namely, after weeks and weeks of testing her and resorting these these FN100 types of caps, she frequently made the comment, well, this cap here, I mean, I'm going to put it right here, but there's something missing from this order because something else needs to be here. This is not complete. Are you sure you didn't lose a cap or something like that, right? So we felt that since she repeated this a few times, that this was maybe a point of singularity in the space where she had a non-monotonicity in the sorting of the caps. Okay. So we tested two background levels, orange and gray, and here's kind of a cheesy mock-up of the little experiment we did. And Ellie compelled me to use Cartman to show you the direction of the motion. That's this thing spinning, and then at some point Cartman doesn't know which direction is going the opposite direction. And then eventually illusory motion is minimized at the point where isoluminance is achieved. And this is the region where the isoluminance setting is obtained for a given subject for all the 20 colors that we tested. Okay. So the results we have for this particular preliminary study, we're going to present to you in equiluminance planes for different observers in an RGB space, because in an RGB space we don't make any assumptions about the LM and S modeling of the particular cone classes that the subjects have. These are just monitor primaries and the DAC values, so it's a very simple, straightforward way of looking at the color. And here we have two subjects plotted, our tetrachromat, who is CA, and an aggregate average normal subject in RGB space. And you know, it's not really easy to see in these particular plots, although you can detect a little bit of a difference over here in this region, showing that these error bars are one standard deviation deviance of the 95% confidence interval for the data, for the data of the staircase. But if we look at a close-up of these regions and we boil it down to an RGB plane, we start to appreciate what the difference is between these different kinds of subjects who have these different phenotypes. So the subject who is the diamond on the left is our potential tetrachromat, and the triangle in the middle is the aggregate subject who is the trichromat phenotype. And the subject over here on this side is the other heterozygote who has the double heterozygosity. Okay? And these are just simply plotted in the R and B DAC values of the space for gray background and for orange. And these are highly significantly different, and it turns out that one of the results that we showed is that there's a whole lot of individual variation, even among normal subjects in these types of settings. But in this particular region of the space, this heterozygote is different quite a bit more. Okay. A better way to look at these data is to simply look at the Euclidean distances between the RGB space parameters and of the MMF equiluminance settings. Okay, so it's a simple Euclidean distance. It's not complicated. So to do that, we take this equiluminance plane, this line that you see in the space, this locus, we snip it at the region I showed here, or any region really, it doesn't really matter. And we stretch it out onto a line and show it to you in this type of display here. If you have an ideal observer who's you have a repeated measure for, so they run the same experiment twice, you know, you're going to get variation in the task, in the actual settings, but in the ideal case, there should be no variation. So this line should plot at 0 for repeated measures within a subject. But what we see in the case of comparing two normal subjects is we see quite a bit of individual variation, and it's not uniform across the color space, which is a new result. It's not really predicted or measured anywhere. And so this is the RGB, Euclidean distance of the RGB parameters and the difference between two subjects' isoluminance settings, so subtracted off for all these points in the space. And this is actually a line that represents a within subject repeated measure. So there's, across the experiment done twice, a given subject will have, on average, this amount of error in their equiluminance settings. Okay. So this shows that within individuals, normal individuals, the differences across space and isoluminance settings are non-uniform, and they vary up to an order of Euclidean distances in RGB parameters of 8. Compare that with the potential tetrachromat against the average aggregate subject. First note the scale up here. Okay, the size of this difference is topping out at around 14, whereas the previous graph between two normal subjects was topping out at 8. And once again, we notice this similar profile of differences where things are more uniform in this middle region of the spectrum, but they deviate a bit at the ends of the spectrum. Okay? If we compare this graph with the graph that has to do with the within subject repeated measure, which is this lower dotted line, and the normal subjects compared with each other, which is the middle line, and then the average normal subject compared with a potential tetrachromat, you can appreciate what the difference is between these particular isoluminant settings across these subject groups. It turns out that the actual result we're seeing is carried by the luminance code. So that's a good thing, because that's essentially what the task is designed to show. And this is one normal subject compared with the potential tetrachromat subject. And what you see is this large difference right here in this central reddish region, where the potential tetrachromat is requiring less red light for isoluminance settings in order to establish isoluminance. So in essence, the threshold for detecting red light at low light levels is lower for this individual. And that's totally consistent with what we would expect from her opsin gene genotype and what we expect to be her phenotype. The other interesting observation is that her curves are crossing over the normal curves, which suggests this isn't just a simple linear transformation that's going on between the mixtures of the luminance contributions of the different cone classes. Okay, so this result resembles the one I described earlier, where two subjects might arrive at different levels and show different patterns of settings, and that's a very interesting and new result. And the other thing we noted was that these subject differences are luminance-based, they're not chromaticity-based. That's good for our confirming the MM isoluminance design and the stimulus that was selected. You have a normal subject's chromaticity diagram of their settings and a potential tetrachromat's chromaticity diagram of her settings, and there's no difference between these. And it turns out if you compare the first potential tetrachromat, who is the artist, with the second potential tetrachromat, who is the non-artist, or has the double heterozygote polymorphism, the order of magnitude of the difference in the scale, again, is shown by the 12 and the 15, shows that she's even more different from the double heterozygote. One reason for this type of result is that the test we designed for this particular artist subject is specifically designed for her opsin gene polymorphism. This subject has a different set of opsin gene polymorphisms, so we expect to see different findings between her and the normal subjects and the other opsin gene, the other heterozygote. Okay. So the whole pattern here is very consistent with what we would expect if things are being driven by these opsin gene polymorphisms. And it's important to note that in this particular data analysis that I'm presenting here, we haven't taken into account any pre-retinal filtering measures, but we do plan to do that in these data. But it's important to note that for the regions that are showing the largest difference here, we don't expect there to be any changes in the results, because pre-retinal filtering is going to mostly affect the S-cone ends of the distribution of this space. Okay, so to summarize, we ran 7 experiments. I presented you data from 2 here. The overall results found that the subject tended to be most different at the lower luminance levels, which was interesting because it was sort of suggested from her art that we saw this and we built it in and found it. At the higher luminance levels, she was not as different, and she was more different in general in these 2 levels, in the area where we have the adaptation state to be congruent with her photopigment kind of singularity that we observed in some sorting task. And that's, the adaptation state is important for like picking out interesting kind of color complementation aspects of her isoluminant settings. So for question 1, independent of our training, is photopigment genotype an important consideration for MM isoluminant settings? Our tentative conclusion is yes, although we'd like to have more subjects to back this up and I think more data is needed, but it does show that the MM isoluminance tax is a nice option to use for testing this potential phenotype. And does life history of art training seem like an important factor in addition to photopigment genotype for MM isoluminance settings? The tentative conclusion is yes, especially when compared to the one tetrachromat that we observed who was an artist. The second tetrachromat seems more similar to the normals in her particular isoluminance settings. Could be a function of the fact that we designed the test to assess CA. We have to know, we have to run the next experiment. But it seems like we have a good evidence that art training makes a difference. So despite needing additional normal controls and factoring and measuring pre-retinal filtering, our results suggest that regions where CA's large differences are seen, for example along wavelengths, are not likely to be explained away by pre-retinal filtering effects. Interpreting these results for artistic structural color relations such as color complements and color balance, and for the detection of color in low light and shadows suggest important implications for tetrachromacy in art. And across light mixture space, MM isoluminance settings are presumed to be due to the first order motion effects that arise in normals, as well as these tetrachromats from cone-based luminance contrast across heterochromatic stimuli. The marked regions where she differs are presumably due to her L prime cone class contributing to what is a luminous cue in the first order motion task, in addition to the usual code from the M, L, and S that normals have. Okay, and where she differed in her settings is consistent with the idea that the extra cone class is contributing a distinctive subjective brightness signal that significantly differentiates her MM equiluminance settings from those found in normal trichromats. That is, the regions where she varied tended to be the ones that had a substantial reddish component, and in some cases reddish and bluish primaries. And in the latter case, because blue contributes a negligible luminance to the actual code, the actual luminance code, we assume that her differences occurring are because of the L prime cone contrast that's provided by the extra cone class. Okay, that's about it. Happy to take some questions, but this is some of her art. I just thought I'd throw this up here for the heck of it. Whatever, regardless of what you think of it, she likes to paint a lot of color in the shadowy regions of her paintings. And this is one of the things we thought would be indicative of her showing a slight difference in the luminance contrast in shadowy regions in dark and light conditions. So I'm happy to take some questions now.
So it was very interesting. I like the use of the minimal motion technique. But now I'm wondering to what extent you were after demonstrating true tetrachromacy as opposed to a kind of anomalous trichromacy. You show that you can detect the very likely presence of this extra cone class. But is her perception truly tetrachromatic? And how easy is it that going to be to detect with a monitor that only has? Yeah. So have you thought of just going on to a color matching experiment with 4 lasers?
This is when I said to you that color tests have been woefully inadequate. I mean not just standardized sorting in pseudo-isochromatic plates, but any test that's done with existing technology is constrained and almost rules out the potential finding of a 4-dimensional color space because they all use 3 primaries. And anytime you use 3 primaries, you run into trouble.
That was the part of my question.
Now here, we're using a luminance code. And above and beyond color, luminance is independent. Luminance doesn't matter what the makeup is of the primaries. It can come from 2 primaries, 3 primaries, 4 primaries, it doesn't matter. So what was detected here was not color changes per se. Changes in the luminance profile when the spectral distribution of the colors is different. So since we have these somewhat cone isolating types of color some of them stimulate S cones more, some of them stimulate M cones more, some of them stimulate L cones more. And then for the cases where L cones were stimulated more, this is where she showed her difference. It's not a difference in color contrast, it's a difference in luminance contrast. So the issue about the 3 guns and the 3 primers is absolutely crucial to designing experiments for this type of subject. You can't find an extra dimension of color unless you have another parameter to vary, right? But in this case, we stood back from color and we said, let's look at a correlated type of phenomenon, which is luminance, and let's look at this highly reliable measure, which is minimal motion, which everybody says is the best way of setting an isomorphic plane for any subject, whether it's dichromatic, trichromatic,
So at one level, you have a method that would let you detect a systematic variation relative to some normal population of normal trichromatic.
Yeah.
But after that, are you interested in going beyond that?
Of course.
Because you may be able to do that with really existing technology. It would be like projectors with, say, filters.
What we have is we
narrow
have
down the light wavelength light, and then you could do the true tetrachromatic matching experiment.
So we do have equipment that was purchased not too long ago that we're in the process of working out that is called an agile light source. It's a cone filtered broadband light source. You have an arc lamp, and it's digital filter projection device where you can filter at the bandwidth of 4 nanometers. So what you can do is you can send a spectrum to the machine, and it will draw you the spectrum. Now what you want to do is and it will project the light, right? So what you want to do is you want to use this primary and find the class of colors that are metamerisms for trichromatic, but which a tetrachromatic can distinguish. And this is the kind of experiment that we would ideally hope to do, and that's coming up. But these technologies are only very recent. I mean, everybody's using RGB monitors and anomoscope matching, which is even a more reduced number of primaries, and all these other things that are designed to detect deviations from trichromacy that are on the deficient end of the tail, not in this other region, right? So they're all designed to detect deviations from trichromacy that are dichromats or anomalous trichromatic or anything down here on the other end of the distribution. So some new methods have to be arrived at, and I've used a few others and have found suggested differences. But with this new type of device and using a higher dimension of primaries, we should be able to show something in color, not just in luminance. So, yes?
So this is fascinating. I'm trying to figure out how the physiology would work for tetrachromacy because, correct me if I'm wrong, would it necessitate that the L cones and the L prime cones would have synapses that modulate the ganglion cells differently? So otherwise, do you end up having, I think the talk is fascinating, don't get me wrong, but do you end up basically having the same input at the level of the ganglion cells with the difference that now this participant has a somewhat broader range of sensitivity?
Has like a different average spectrum in the L range, right?
As an excellent question, both excellent questions, by the way. The first issue that you have to recognize is that E lambda for these subjects is expected to be different. That's the first step.
Say that again?
E lambda, so the photopic luminosity function, the average response to daylight is expected to be different just by standard models, okay? Just like a dichromat, a colorblind person's E lambda is different, okay? The second thing is, not really anything is known about what the coupling is in the opponent process mechanism of the LGN. There's a theory that it's red couples with green and yellow couples with blue, and this whole thing happens, right? And this is the theory. But they find with people with highly biased retinas, that is, there's a lot of variation in the mosaic that occurs. And you can get people that have predominantly red cone retinas and predominantly M cone retinas, and their whole range in between. And so there's some interesting type of neural wiring going on that's highly individual variant, And this is very common. Okay, now, to answer your question about the possible organization of the opponent signals, because of the similarity between the M and the L opsin genes, they only differ on 15 sites. They're so homologous, the actual genes, that the cells are almost, they actually are a duplication. So one arose from a duplication of the other, okay? They only differ on 15 sites on 3,000 base pair sequence. So what happens frequently is there's tons of crossover in meiosis, okay? So you have a whole range from a pure M to a pure L and everything in between, where the components are weaving themselves together. So you have the whole, I had a slide in here, but I took it out. You have a whole range of possibilities, right? Every single slot is possibly substituted. What this means is that the phenotypes out there in the world, they're not just M and L. They are trichromats, but they have M1, M2, or M3, or M4, or M5, and they have M1, L1, or L2, or L3, or all these variations. And not only that, but everybody here in this room who's a trichromat has multiple copies of these genes in a head-to-tail array, so that if one drops out, the other one pops in. So in terms of the population phenotype, it's a real misnomer to say that there is a trichromat standard in the population, because there's so much variation. These individuals that have this obligate extra chrome type have been shown to express them in their retinas. So this is called weak tetrachromacy. We know the code's in the retina, okay? Whether they actually hook up into these opponencies between an L prime and an M, or an L prime and an L, that's entirely within the realm of possibility, and stuff like that happens in non-human primates. So that is known, okay? So it's just not verifiable at the present point. You can do adaptive optics to image the retinas, but they can't resolve that 5 nanometers yet. They can resolve 30, but they can't resolve the 5. So as soon as they get the adaptive optics down to a level where they can tell the difference between the two L prime, the O prime and the L, then we'll know a lot more about the actual populations of the retinal mosaics and all these other things. We'll be able to do a little bit more on the neural wiring of